Hello everyone, good day. We are the Group H. This discussion covers the whole chapter 33, which is all about nervous system and sense organs. So before we are going to go over with the nerve signals, we must know first the function of nervous system. So what is the function of nervous system? So the nervous system is a rapid communication system that interacts continuously with the endocrine system to control coordination of the body function. So nervous system is one of the unique organ in our body that enables the body to respond and adapt to the changes both inside and out. So the basic unit of nervous integration in all animals is neuron. Neurons are highly specialized cells designed to conduct self-propagating electrical events called action potentials to other cells. 99% of neurons in the body lie entirely within the central nervous system. Neurons are connected to the receptor that converts the stimuli into nerve signals. The nerve cell is composed of nerve terminals, axons, dendrites, nucleus, cells including Schwann cell. So the function of dendrites is to receive communication from other cells, while axons carries nerve impulses away from the cell. So it is also called nerve cell. It is very helpful in transmitting the information to the nervous system. So we also have here the Schwann cell. So myelinate the axons of P and S, myelin. It is the fatty layer that includes the axon and helps to increase the conduction of neurons. So the nerve terminal, so it is a specialized region of a neuron that releases neurotransmitters when stimulated by an electrical signal from axon. So this is an image of a Schwann cell that surrounds the axon. Myelin composed of multiple layers of glial cell plasma membrane that wrap around the axon in concentric rings as it forms the myelin sheath. Myelin is produced by a special glial cell called Schwann cell. So action potential. What is an action potential? So it is an electrochemical message of neurons, the common functional denominator of all nervous system activity. It is very rapid and brief moving change in electrical membrane potential that results from a dipolarization of a membrane of the nerve fiber. So action potentials are transmitted from one neuron to another across synapses, which may be either electrical or chemical. So the narrow gap between neurons at chemical synapses is bridged by chemical neurotransmitters molecule.
Synapses, junctions between neurons. Synapse, a small gap where an action potential must cross before reaching the other neuron, separating neuron from another neuron. Axon terminal, also known as synaptic knob or synaptic terminal, branches at the end of the axon that interacts with a dendrite or the cell body of the next neuron. Synaptic cleft, a narrow gap between two neurons. A small gap between the axon terminal of the presynaptic neuron and the membrane of the postsynaptic cell. Presynaptic neuron, neurons bringing action potential towards chemical synapses. The firing of an action potential in one neuron or sending neuron causes the transmission of a signal to another neuron. Postsynaptic neuron, carrying action potentials away. Neuron that receives the neurotransmitter after it has crossed the synapses. Electrical synapses, allowing direct ion current transfer between cells to pass the action potential to the synaptic cleft. Unlike the chemical synapses, there is a direct physical connection between the presynaptic neuron and the postsynaptic neuron. This connection takes the form of a channel called a gap junction, which allows current ions to flow directly from one cell to another. Electrical synapses transmit signals more rapidly than the chemical synapses do. Some synapses are both electrical and chemical. At these synapses, the electrical response occurs earlier than the chemical response. One benefit for electrical synapses is fast signaling. Example, defensive reflex in animals. Chemical synapses. Type of synapse with wider, more complex and slower signaling synapse. Chemical transmission involves release of the chemical messenger known as neurotransmitters. In a chemical synapses, when an action potential or nerve impulse arises at the action terminal, it activates voltage-gated calcium channels in the cell membrane. Ca2+, which is present at much higher concentration outside the neuron than inside, rushes into the cell. The Ca2+ allows synaptic vesicles to fuse with the axon terminal membrane, releasing neurotransmitters into the synaptic cleft. The molecules of the neurotransmitter diffuse across the synaptic cleft and bind to receptor proteins of the postsynaptic cell. Activation of the postsynaptic receptors leads to the opening or closing of ion channels in the cell membrane. This may be depolarizing, making the inside of the cell more positive, or hyperpolarizing, making the inside of the cell more negative, depending on the ions involved. Neurotransmitters are often referred to as the body's chemical messengers. They are the molecules used by the nervous system to transmit messages between neurons or from neurons to muscles. Synaptic vesicles. These are membrane-bound spheres filled with neurotransmitter molecules. One of the most common neurotransmitters of the peripheral nervous system is the acetylcholine. Chemically gated channels. Channels in the postsynaptic membrane that open and close response to chemicals such as neurotransmitters. Excitatory synapses. Depolarization. A synapse in which the nerve impulse in a presynaptic cell increases the probability of the postsynaptic cell to fire an action potential. It makes the inside of the cell more positive, bringing the membrane potential closer to its threshold for firing an action potential. Inhibitory synapses. Hyperpolarization. A kind of synaptic potential that makes a postsynaptic neuron less likely to generate an action potential. Tends to keep the membrane potential of the postsynaptic neuron below the threshold for firing an action potential.
Now we'll be discussing the evolution of nervous system. In vertebrates, the development of centralized nervous systems. Animal phyla reveal an increase in the complexity of nervous systems that reflect, in a general way, stages in the evolution of the nervous system. Simplest pattern of invertebrate nervous systems are the nerve net of radially symmetrical animals such as sea anemones, jellyfishes, hydras, and comb jellies. So these are various invertebrate nervous systems. We see the nerve net of the hydra, the two main nerve trunks of the flatworm, the bilobed brain and segmented ganglia of the earthworm, and the grasshopper, which have a similar nervous system as the annelids but have larger ganglia. The nerve net is an extensive network in and under the epidermis all over the body. A signal starting in one part of this net spreads in all directions, since synapses in most radially symmetrical animals allow bidirectional signal transmission. There is an evidence of organization into reflex arcs. Now, many responses are astonishingly complex for such a simple nervous system. A cnidarian's nerve net is often concentrated into two nerve rings into their medusan forms. They receive sensory input from statocysts, organs of balance, ocelli, light-sensitive organs, and sensory cells detecting chemical and tactile stimuli. We can see the cnidarian's nerve rings here. After the nerve net, there is the bilateral nervous systems. They represent a distinct increase in complexity over the nerve net of radiate animals. Flatworms often have this nervous system. They have two anterior ganglia from which two main nerve trunks run posteriorly, with lateral branches extending throughout the body. This is the simplest nervous system, showing differentiation of a peripheral and a central nervous system. This bilateral nervous system is seen here. In annelids, they have an elaborate nervous system that consists of a bilobed brain, a double nerve cord with segmental ganglia, and a distinctive afferent and efferent neurons. Segmental ganglia are relay stations for coordinating regional activity. This pair of cerebral ganglia represent the bilobed brain, and the ventral nerve cord has segmented ganglia. Mollusks have a nervous system that has a basic plan of 3 pairs of ganglia and 2 pairs of nerve cords. Cephalopods have ganglia burgeoned into textured nervous centers of great complexity. Octopus nervous systems may contain more than 160 million cells. They have highly developed sense organs, they are capable of learning, and they possess the largest brain of any invertebrate. This is a ventral view of the brain of a vampire squid. Arthropods have a basic nervous system that resembles that of annelids. However, their ganglia are larger and sense organs are much more developed. Their social behavior is often elaborate. They are capable of considerable manipulation of their environment. Examples of this learning have been documented in bees, wasps, ants, flies, locusts, and grasshoppers. There's a brain region associated with learning in areas called mushroom bodies, seen here in the right. They are largest in social insects, and experiments have shown changes in these structures have been observed with age and experience. Now we'll be moving on to vertebrates and the fruition of encephalization. The basic plan of the vertebrate nervous system is a hollow dorsal nerve cord terminating anteriorly in a large neural mass, or the brain. This pattern contrasts with the nerve cord of bilateral invertebrates outside the phylum Chordata, where their nerve cord is solid and ventral to their alimentary canal. Encephalization is by far the most important trend in the evolution of vertebrate nervous systems. It features the increase in size, configuration, and functional capacity of the brain. It brought to full fruition several functional capabilities such as fast responses, great capacity for storage of information, enhanced complexity and flexibility of behavior. They gave the ability to form associations between past, present, and in humans, future events. So a group of neurons that work together is called the reflex arc, the fundamental unit of neural operation that has remained conserved during the evolution of the nervous system. It contains at least two neurons. The parts of a typical reflex arc are a receptor, an efferent, a central nervous system, an efferent, and an effector. In its simplest form in vertebrates, it contains only two neurons, a sensory neuron and a motor neuron. An example of this is the knee-jerk or stretch reflex. As you can see, this is the stretch reflex, wherein sudden pressure on the patellar ligament stretches muscles in the upper leg. In reflex arcs, usually interneurons are interposed between sensory and motor neurons. An interneuron may connect afferent and efferent neurons. A response to a stimulus acting over a reflex arc is called a reflex act and is mostly involuntary. Examples are with many vital processes of the body such as breathing, heartbeat, diameter of blood vessels, and sweat secretion. Some acts are innate, while others are acquired through learning. In almost every reflex act, a number of reflex arcs are involved. A single afferent sensory neuron may make synaptic connections with many efferent motor neurons. An efferent motor neuron may receive signals from many efferent sensory neurons, and reflex activity may also be modified by signals from the brain. The central nervous system, or CNS, is composed of the brain and the spinal cord. During early embryonic development, the spinal cord and brain begin as an ectodermal neural groove, which by folding and enlarging becomes a long, hollow neural tube, which is shown in the diagram on the right. The cephalic end of this neural tube enlarges to form brain vesicles, and the rest become the spinal cord. The spinal cord is unlike any invertebrate nerve cord, as it has segmental nerves of vertebrate spinal cords, 31 pairs in humans, which are separated into dorsal sensory roots and ventral motor roots. Sensory nerve cell bodies are gathered together into dorsal root ganglia. Both dorsal and ventral roots meet beyond the spinal cord to form a mixed spinal nerve. The spinal cord encloses a central spinal canal and is additionally wrapped in 3 layers of membranes called meninges, shown here. In the cross-section, the cord shows 2 zones, an inner zone of gray matter, resembling the shape of the wings of a butterfly, containing the cell bodies of motor neurons and interconnecting interneurons, and an outer zone of white matter, which contains bundles of axons and dendrites linking different levels of the cord with each other and with the brain. Unlike the spinal cord, which has changed little in the structure during vertebrate evolution, the brain has changed dramatically. The ancestral vertebrate brain of fishes and early tetrapods expanded to form a deeply fissured and enormously intricate brain in the lineage leading to mammals. It reaches its greatest complexity in the human brain, which contains some 35 billion neurons, each of which may receive information from tens of thousands of synapses at one time. So, it is shown here the evolution of the vertebrate brain, from the lamprey all the way to a horse. Note the progressive size of the cerebellum. The cerebellum, which is concerned with the equilibrium and motor coordination, is largest in animals whose balance and precise motor movements are well developed. See the difference between a horse and a frog cerebellum. The ratio between the weight of the brain and that of the spinal cord affords a fair criterion of an animal's intelligence. In fishes and amphibians, this ratio is approximately 1 to 1. In humans, the ratio is 55 to 1. In other words, the brain is 55 times heavier than the spinal cord. Although the human brain is not the largest, the sperm whale's brain is 7 times heavier, nor the most convoluted, that of the porpoise is even more fluid. It is the best in overall performance. Brains of early vertebrates had three principal divisions: a forebrain, a midbrain, and the hindbrain. These primitive but very fundamental concerns of the brain have been in some instances amplified and in others reduced or overshadowed during continued evolution as sensory priorities were shaped by an animal's habitat and their way of life. The hindbrain has the medulla oblongata, or medulla, the most posterior division of the brain. It's really a conical continuation of the spinal cord. The medulla, together with the anterior midbrain, constitutes the brainstem. The brainstem is the area that controls numerous vital and largely subconscious activities, such as the control of the heartbeat, respiration, vascular tone, gastric secretions, and swallowing. This also contains centers that appear to integrate incoming peripheral information regarding satiety and feeding stimuli. The pons is also a part of the hindbrain. This contains a thick bundle of fibers that carry impulses from one side of the cerebellum to the other. It also connects both medulla and cerebellum to other brain regions. Another part of the hindbrain, called the cerebellum, lies dorsal to the medulla. This controls the equilibrium, posture, and the movement of the body. The development of the cerebellum is directly correlated with the animal's mode of locomotion, agility of limb movement, and their balance. Due to this, the cerebellum is usually weakly developed in amphibians and non-avian reptiles, which are forms that live close to the ground. They are well developed in more agile bony fishes, and they are most highly developed in birds and mammals, in which it is greatly expanded and folded. It does not initiate movement, but rather operates as a precision error control center, or servo mechanism that programs a movement initiated somewhere else, such as the motor cortex of the cerebrum. Primates, and especially humans, who possess a manual dexterity far surpassing that of other animals, have the most complex cerebellum. Movements of hands and fingers may involve the cerebellar coordination of simultaneous contraction and relaxation of hundreds of individual muscles. The midbrain consists mainly of the tectum, including the optic lobes. They contain nuclei that serve as centers for visual and auditory reflexes. They have undergone little evolutionary change in structure among vertebrates, but has changed markedly in function. The midbrain mediates the most complex behavior of fishes and amphibians, integrating visual, tactile, and auditory information. Such functions have been gradually assumed by the forebrain in amniotes. In mammals, the midbrain is mainly a relay center for information on its way to higher brain centers. Now onto the forebrain. At the anterior of the midbrain lie the most posterior elements of the forebrain. These are the thalamus, which is an egg-shaped major relay station, which analyzes and passes sensory information to higher brain centers. The hypothalamus, which has several housekeeping centers that regulate the body temperature, water balance, appetite, and thirst. A hypothalamus functions are mainly concerned with maintenance of internal constancy, or homeostasis. Neurosecretory cells located in the hypothalamus produce several neurohormones. It also contains centers for regulating reproductive function and sexual behavior, and it participates in emotional behaviors. The anterior portion of the forebrain, or cerebrum, can be divided into two anatomically distinct areas, the paleocortex and the neocortex. Originally concerned with smell, the paleocortex became well developed in advanced fishes and early terrestrial vertebrates, which depend on this special sense. In mammals, and especially in primates, the paleocortex is a deep-lying area called arenencephalon, also called the nose brain, because many of its functions depend on olfaction. Better known as the limbic system, it mediates several species-specific behaviors that relate to fulfilling needs, such as feeding and sex. One region of the limbic system, the hippocampus, has been extensively studied as a site of spatial learning and memory. The hippocampus is now well known because its neurons have mitotic capabilities in adults, a previously unknown occurrence in mammalian neurons. The neocortex completely overshadows the paleocortex and has become so expanded that it envelops much of the forebrain and all of the midbrain. almost all integrative activities primitively assigned to the midbrain are transferred to the neocortex, or in other words, cerebral cortex, that forms the surface gray matter of the cerebrum. The right and left hemispheres of the cerebral cortex are bridged through the corpus callosum. In humans, the two hemispheres of the brain are specialized for different functions. The left hemisphere is for language development, mathematical and learning capabilities, and sequential thought processes. The right hemisphere is for spatial, musical, artistic, intuitive, and perceptual activities. Each hemisphere controls the opposite side of the body. Hemispheric specialization has long been considered a unique human trait, but it was recently discovered in the brains of songbirds, in which one side of the brain is specialized for song production. So here are the parts of the human brain. We see here the medulla oblongata, and together with the spinal cord is the brainstem. Here we have the pons. We have here the cerebellum. The thalamus is over here. The hypothalamus is also here, the midbrain, and here is the corpus callosum, which connects the two hemispheres. Now we're moving on to the peripheral nervous system, which includes all nervous tissue outside the central nervous system. There are two functional divisions: the sensory or afferent division, which brings sensory information to CNS, and the motor or efferent division, which conveys motor commands to muscles and glands. The efferent division has two components: the somatic nervous system, which innervates skeletal muscle, and the autonomic nervous system, which innervates smooth and cardiac muscle and glands. The autonomic nervous system governs involuntary internal functions of the body. This includes the movements of the alimentary canal and the heart, contractions of smooth muscle of blood vessels, the urinary bladder, the iris of the eye, secretions of various glands, and others. The autonomic system is further divided into the sympathetic and parasympathetic systems. The parasympathetic division is mostly associated with non-stressful activities, such as resting, eating, digestion, and urination. The sympathetic division is active under conditions of physical or emotional stress, conditions such as increase of heart rate, dilation of the blood vessels towards the skeletal muscles, constriction of the blood vessels in viscera, activity of intestinal tract decreases, and the metabolic rate increases. This is the general organization of the autonomic nervous system.
So, sense organs. Sense organs, every animal requires this for a constant information from environment to regulate their lives. Sense organs are specialized sensory receptors that have evolved to take environmental status and change. An animal's sense organ are its first level of environmental reception to bring information to the central nervous system, which is the whole reason why animals don't die, because they absolutely never realized there was a tree in front of them. That is why a lot of animals would have died without any sense organs. The sympathetic and parasympathetic nervous system. For the sympathetic nervous system, that is anything that is to increase or dilate so you can see better your eyes, stop secretion of glands so you don't salivate, dilate bronchioles so you can take in more oxygen, speed up heartbeat so you can give oxygenated blood throughout the body more efficiently, secrete adrenaline for the adrenaline gland, that's its entire purpose, decrease secretion of the stomach to maintain water and nutrients within the body, decrease motility so you don't poop during any stressful situation. Then retain colon contents. So you also want to keep the nutrients and not poop in the middle of a fight or running. And delay stimulus. Stimulus is some form of energy, electrical, mechanical, chemical, or radiant. This is a sensory receptor within a sense organ transform energy from a stimulus into a nerve action potential, which is the common language of the nerve system. Sensory receptors are, as a rule, specific for one kind of stimulus. Example, eyes to light, ears to sound, pressure receptors to pressure, and chemoreceptors to chemicals. Because you don't taste with your fingers, and you don't touch with your tongue. You kind of can, but law of specific nerve energies, which is explained type of that. The real perception of sensation is performed by localized regions of the brain, where sensory receptors from each sense organ have their own hookup. So then that is just a summary of why you can only touch with your touch, you can only taste with your tongue, you can only see light with your eyes, and you can only smell with your nose. This concept is by Johannes Müller in 1830s, which is the concept of labeled lines. They are labeled to exteroreceptors, those near the external surface that keeps the animal informed of the external environment. Interoceptors is the internal part of the body which receives stimuli from internal organs. Proprioceptors, found in muscles, tendons, and joints that provide an organism with a sense of body position. And for the last one is sometimes receptors are classified by a form of energy. We classify them into chemical, mechanical, light, or thermal. Chemoreception is the oldest and most universal sense in the animal kingdom, and in unicellular eukaryotes probably guides organism behavior more than does any other sense. Unicellular forms use contact chemical receptions to locate food and adequately oxygenated water and to avoid harmful substance. These are receptors elate an orientation behavior called chemotaxis, which is towards or away from a chemical source, towards if it's food, away if it's predators. And the top one, chemoreception, is just basically, why lions piss to have their term. Distance chemical receptions, which are often developed to a remarkable degree of sensitivity, because it has to be really sensitive, like the smell olfaction or the nose of dogs. They have to smell something from afar, because that's the entire purpose of their survival. That's one of their purpose of survival, is to smell something from far away. It's usually smell or fashion for feeding behavior, location, selection of sexual mates, territorial and trails marking for some ants, and alarm reaction of numerous animals. In all vertebrates and in insects, the sense of taste and smell are clearly distinguished, because we know soap smells good, but we also know if you ever taste it, soap tastes bad. So they're very distinguished. Then for insect chemoreceptors are located in sensory hairs called sensilla. Taste sensilla occur on the mouth parts for taste, and then their legs. Pheromones. Social insects and many other animals, include mammals, produce species-specific compounds that constitute a highly developed chemical language. Because pheromones, basically, if you can't smell it, then you're very not developing chemical language. Just like if animals don't make sounds, they're very not specialized in vocal, so in vocal language. Taste buds consists of a cluster of receptor cells surrounded by supporting cells and has a small external port through which slender tips of sensory cells project, aka your tongue. It has taste buds so you can taste. There are 5 basic tastes. There's sweet, sour, bitter, salty, and umami, which is savory. Many terrestrial vertebrates possess an additional olfactory organ, the vomeronasal organ or Jacobson's organ. Food odors for snake and pheromones for mammals. For snakes, that's why they move their tongue out and bring it back in. It's tasting the air for food. Kinda like tasting air for food. Mechanoreception: touch, pressure, stretching, sound, vibration, gravity. In short, they respond to motion. Mechanicals. Touch. Each hair follicle is crowded with receptors sensitive to touch. Touch-sensitive spots on the surface of your body most occur in the tongue for tasting, and fingertips, which is why fingers first. Then the Pachynia and Corpuscle, relatively large mechareceptors that register deep touch and pressure in mammalian skin. Just common deep layers of skin, connective tissue surrounding muscle, tendons, abdominal movements. Just remember the word deep touch. If there's something touching very deep with high pressure in your body, you'll feel. Mechanoreception or pain. Pain receptors, unspecialized nerve fibers ending that respond to a variety of stimulus signaling. Possible or real damage to tissues, which is the whole purpose of pain. Oh, it hurts. It could be something harmful, or oh, it hurts. I am already damaged. Please save me. For pain fibers respond to small peptides such as substance P and bradykins, which are released by injured cells, aka a part of your body is just kind of like, bro, you've burned me enough times already. Can you save me for once, please? And that is slow pain. For fast pain, this is response for something fast. It's pain prick, or cold or hot stimuli, or a more direct. It's a more direct response to the nerve endings. Thermal stimulus or stimulant. It's like, oh, it's hot. Ow! You don't do that. You go, oh oh, it's hot. And that's why it's faster. And then endorphins and echinophilia are morphine-like opioid activity. When they release, they bind with specific opiate receptors in the midbrain. Just remember their body's own analgesic or pain reliever. You break a finger while running away from something in pure panic, you probably won't feel it. But once you're in the hospital, you'll be crying, probably. Yeah. Mechanoreceptor for fish. Lateral line is a distant touch. Just read that. Neuron mass are located in the body surface and aquatic amphibians and some fishes. Each of us is a collection of hair cells with sensory ending or cilia, hair, embedded in a gelatinous wedge-shaped mass called the cupula or cupula in the bottom right. So basically, it's a way for fish to understand that this pressure change here. Move away from it, yeah? For mechanoreceptor hearing, an ear is a specially set up for detecting sound waves in the surrounding environment. Very new, wow. Most invertebrates are actually inhabiting a silent world. So only certain arthropods, these have developed true sound receptor organs. And even among insects, only few have it. So, yeah, just you kind of feel bad for some of them, yeah? Mechanoreceptors. This is in the lab, the ear has a labyrinth, and then it's a fluid-filled cavity. But it's just the main point is it starts with the external auditory canal for catching the sound, tympanic membrane, the eardrum, then there's three ossicles, the hammer, the anvil, the stapes. You see in the picture there's three ossicles. Then the oval window, which is going to the snail shell structure. Then turbulence to the fluid labyrinth, then to the hair cell and the organ corti is stimulated, which is interpreted by the brain to be equilibrium. The vertebrate organ equilibrium is the labyrinth or principal organ, consists of two small chambers, the saccule and utricle, and three semicircular canals as seen at the top right. Fluid found in the semicircular canal is called the endolymph. The utricle and the saccule are static balance organ, basically balanced, like statuses. It's photoreceptor vision, a light-sensitive receptor. Two different eyes, there's single lens, simple eyes, and multifaceted comparas. Comparas is composed of many independent visual units called omatida. The spherical eyeball is built of three layers: the outer white sclera for support protection, a mirror choroid coat, and then there is the light-sensitive retina. Then the cornea is a transparent anterior modification of the sclera. The pigmented region or iris regulated for the size of the light opening. Lens are a transparent elastic oval disc that bends slightly to focus an image to the retina. Ciliary muscle attached to and encircle the lens to alter the curvature of the lens so that image varying distance can be focused on by the retina. Between cornea and lens is an outer chamber filled with watery aqueous humor. For the inner chamber between the lens and the retina is called the outermost layer, closer to the sclera, consists of pigment cell or chromatophores. Adjacent to this layer are the photoreceptors, rods, and cones. Cones are more concerned with color vision in ample light, and rods have colorless vision in dim light. Fovea centralis or fovea, the region of keenest vision, is located in the center of the retina. Chemistry of vision. Both rods and cones contain light-sensitive pigments called rhodospins. Rhodospins molecule consists of a large protein, diopsin, an enzyme, and a small carotenoid molecule, retinal, a derivative of vitamin A. Color vision. Cones function to perceive color and require 50 to 100 times more light for stimulation than do rods. Night vision is almost totally rod vision. Unlike humans, we have both day and night vision. Some vertebrates specialize for one or the other, strictly nocturnal animals such as bats and owls have pure rod vision. Purely diurnal forms or day animals such as common gray squirrels and some birds have only cone vision, which means they're virtually blind at night.